In this module, we will talk about the last range of fermentation process. We can say that the scope of fermentation process, in which we will talk about the recombinant products which we can produce through fermentation process. In actual sense, when we talk about the recombinant products, that can also come in first 4 ranges as concerned biomass production, metabolite production, biotransformations, and enzyme productions. What are the recombinant products? Basically, the recombinant products and the involvement of fermentation process is based, you can say that this story is whole based upon with the advancement of the modern biotechnology. Or we can say that with the advent of recombinant DNA technology. Now, we know that with this technology, we can utilize the microorganism, their expression system. We can produce different products by using the foreign genes incorporated into the microorganisms. So, if we use first the recombinant DNA technology or a molecular biology techniques just to have genetically modified organisms, then by using those genetically modified organisms, by their mass culturing, we can produce their biomass for getting a more DNA, which we call as a recombinant DNA. On the other hand, we can produce different metabolites, which we can are the recombinant metabolites. Basically, those metabolites are not basically produced in those microorganisms. We carry those genes from higher plants or from higher animals in which there is difficult to produce in a large scale. There are various examples we can see in the next slide, but here the recombinant products are basically those which we can utilize by the use of molecular biology techniques. So, in this slide, you can see that different types of recombinant products. As an example, interferon. Interferons, we know that is mostly used in the treatment of hepatitis. So, by the use of the modern biotechnology, we incorporate those genes into E. coli and then produce some interferons. As concerned the insulin, and we know that mostly in older times the insulin is extracted from the liver of the animals. But now with the advent of recombinant DNA technology, we clone those genes in E. coli or in a Pichia pastoris, and then by the mass culturing of those, we can produce industrially, and insulin is now producing through fermentation process. And third example is human serum albumin, and the fourth is clotting factor 8 and 9, and then epidermal growth factors, calf chymomycin, and bovine somatostatin. So these are different recombinant products which is already industrialized through fermentation process. Up till now, we have covered different ranges of fermentation process. Now I will talk about that what is the chronological development in fermentation industry. So we know that the fermentation subject, or a fermentation as a discipline, or a fermentation as a technology is a very conventional and a very old type of science. We say that the modern biotechnology also based upon fermentation technology. If we draw the, we can say that if we draw the time scale of the fermentation technology or a fermentation industry development, we divide into 5 different eras. Before 1900, and then 1900 to 1940, and 1940 to date, and 1964 to date, and 1979 to date. So as concerned the first era, the mostly 2 products were very common during the fermentation process. One is alcohol, beer and wine, and then is vinegar. But after 1900 to 1940, baker yeast, glycerol, citric acid, lactic acid, acetone, or a butanol-like products formed. But in third era, 1940, then new products, penicillin, streptomycin, gibberellin, amino acids, nucleotides, and number of enzymes start producing through fermentation process. But in the fourth era, which started from 1964 to date, in which the single cell protein using hydrocarbons and other feedstock along with the previous products become commercialized through fermentation process. But in the modern era, we can say that start from 1979 to date, that is the production of heterologous protein by microbial and animal cells, MABs production by animal cells, and so many recombinant products which I already discussed in previous slide. So as concerned the product, and now I will talk about the different, you see that different fermentation process with respect to chronological development from pre to 1900. Mostly the batch system was used, and then from 1900 to 1940 most of the product were formed through batch and continuous process. But nowadays, that from 1942 onward, three types, batch, continuous and fed batch systems are used.